Diphtheria is a potentially fatal and highly contagious disease caused by Corynebacterium diphtheria. It is spread by aerosol transmission. Nowadays in England, most people have received the diphtheria vaccine, so this disease does not pose a current threat. The bacteria is a small, non-motile, gram-positive, non-spore-forming rod-shaped organism that does aerobic respiration and has 4 subspecies. The bacteria most commonly colonizes the upper respiratory tract of the human host, although colonization of other anatomical areas such as the skin is possible. Its action in the upper respiratory tract leads to the formation of white inflammatory pseudomembranes. These pseudomembranes are composed of dead epithelial cells and inflammatory cells such as neutrophils. In severe cases, the pseudomembranes can obstruct the airway, leading to death by suffocation. But what are the key virulence factors that allow the bacteria to cause such damage to the epithelium of the upper respiratory tract? In fact, there is only one virulence factor that is responsible for the tissue damage seen during a diphtherial infection. This factor is the diphtheria toxin, a secreted, simple AB toxin, which will now be referred to as DT. DT is so fascinating not only because it is considered the prototypical simple AB toxin, but also because it is used nowadays as part of an anti-cancer compound. Let's have a look closer at its structure, mechanism of action, and use in cancer therapy. As mentioned, DT is a simple AB toxin, which means it is composed of an A and B subunit. Like all simple AB toxins, the A subunit is the enzymatic portion, while the B subunit is the portion responsible for toxin binding to the target cell surface. These two subunits are held together by a critical disulfide bond, and as we will see later, reduction of this bond is a crucial step in allowing the toxin to cause damage to the host cell. As well as having two subunits, the toxin can be described as being composed of 3 functional domains, with distinct and important roles. It has an N-terminal catalytic domain, a C-terminal receptor binding domain, and a translocation domain in between, the precise role of which will become clear soon. The domains can be referred to as the C, R, and T domain respectively. Now let's have a look at the life of the toxin. It is synthesized and secreted from the bacteria as a single polypeptide chain, but encounters furin peptidases on the surface of host cells. These cleave the chain into the A and B subunits, which then become joined by a disulfide bond between cysteine residues in the 2 portions of the toxin. The R domain then binds its cognate receptor on the host cell. This is heparin-binding epidermal growth factor, or HBEGF for short. Binding is assisted by CD9, a co-receptor also found on the surface of the target cell. This interaction between the R domain and HBEGF triggers uptake of the toxin into vesicles by a process known as clathrin-mediated endocytosis. The toxin is transported in the vesicle to the early endosome. The lumen of the early endosome is very acidic. The acidity here causes a conformational change to occur in the T domain of the toxin, such that it inserts into the endosomal membrane. The catalytic A subunit now gets exposed to the cytosol, but at this point it is still covalently attached to the rest of the toxin. The next step involves the reduction of the critical disulfide bond we spoke of earlier. This is done by cytosolic host proteins, and as a consequence, the catalytic subunit becomes fully activated and free in the cytosol, and from here it can act to bring about cell death. How does it do this? What is the reaction that this subunit catalyzes? It functions to ADP ribosylate, a protein that is vital for the elongation step of protein synthesis. This protein is known as EF2, and the modification made by the toxin to EF2 stops it being able to interact with RNA during translation. Because of this, translation in the cell virtually grinds to a halt, and the cell then dies by apoptosis. It is fascinating to consider that only 1 molecule of DT is enough to kill a human cell. This incredible potency of DT made scientists think that perhaps it could be used to selectively kill cancer cells. They had the idea that if the toxin's binding specificity could be altered, it could be used as a magic bullet that kills cancer cells and leaves healthy ones unharmed. This, of course, was an exciting prospect, seeing as many existing cancer therapies have major off-target actions. All these ideas led to the construction of the immunotoxin. This is a synthetic compound with two portions. One portion is the toxic portion, which is made of the A subunit of DT. The second portion is the binding portion, which is composed of a molecule derived from the immune system. The binding portion can be an antibody, antibody fragment, or a ligand like interleukin 2. These binding portions are chosen because they bind to receptors strongly expressed on cancer cells and almost absent on healthy cells. The immunotoxin is injected into the blood of the patient or directly into the tumor mass, and due to the selection of the binding portion, it will only bind the cancer cells. Like with the natural toxin, binding triggers uptake into the cell, and once here, the toxic portion can prevent translation and bring about apoptosis. To date, immunotoxins have been made using bacterial toxins other than DT and even the plant-based toxin ricin. Many immunotoxins are currently undergoing clinical trials, and there is hope that they will soon be used for the treatment of a variety of cancer types. There is one FDA-approved immunotoxin called ONTAC that is used to treat cutaneous T-cell lymphoma. ONTAC is in fact a DT-based immunotoxin, and there are several other DT-based compounds under investigation. The binding portion of ONTAC consists of IL-2. The transformed T cells that circulate in leukemias and lymphomas such as cutaneous T-cell lymphoma show enhanced expression of the IL-2 receptor, while other circulating white blood cells do not. This is the reason for the selection of IL-2 as the binding portion of the immunotoxin. In summary, we have seen that the diphtheria toxin is an incredibly potent toxin. We have learned about how it binds to and enters a cell, and then how it gains access to its cytosolic substrate. We have seen that the way it causes damage to a cell is by disrupting translation, which in turn brings about apoptosis. Importantly, we have seen that this toxin is nowadays used for human benefit as part of an approved anti-cancer immunotoxin. Although ONTAC is the only approved immunotoxin, this field of research is by no means quiet. Hopefully in the near future we will see more DT-containing compounds being used in the clinic for the treatment of many cancer types.